First of all, I want to say thank you for the kind introduction, Andreas, and also for selecting me for this prize. I'm honored and humbled and everything. And also, I want to say congratulations also to Richard, who's I think also a very worthy awardee for this award. Right, as Andreas said, what we're interested in is systemic lupus erythematodes, which is one of the more common autoimmune diseases in rheumatology. And it is named lupus because people have this typical facial rash which some people reminded of a wolf. Lupus can involve basically any internal organ, and that's what's making it such a challenge to treat lupus and to recognize lupus. Some of the more common manifestations involve inflammation of the skin, so dermatitis, inflammation of the joints, so arthritis, and what's very bad for the patient is the inflammation of the kidneys, the nephritis, which is often the manifestation that leads to loss of life or organ. And as you can see, still in 2017, the 10-year survival of these mostly pretty young women is only 90%. So there's still a large need for additional and better treatments. We understand some parts of the etiology and the pathogenesis of SLE. For example, there are genetic factors, there are environmental risk factors like smoking or UV light, and certainly also hormonal factors, because as I said, 90% of SLE patients are female. All these factors then combine to lead to basically an adaptive immune response. So you have antibodies, autoantibodies against nuclear antigens, and specifically antibodies against double-stranded DNA. And also you have antigen-specific T cells that contribute to the organ damage. Cytokines, so the mediators of inflammation, are, as in any adaptive immune response, also involved. And the hallmark cytokines, one of the hallmark cytokines of SLE, are the type 1 interferons. So cytokines that are typically associated with antiviral response. And all these factors then contribute to organ damage in different organs. Some cells or some mechanisms more in one organ and some more in the other. The focus of, or one of the focuses of that has been put on the pathogenesis of SLE at the DFZ is the contribution of plasma cells. So when a B cell becomes activated, some of the B cells can become long-lived plasma cells and sit, for example, in the bone marrow, but also in the inflamed organs themselves, where they sit in dedicated niches. They don't proliferate anymore. They don't do much, and that's actually a problem because that makes it very hard to target these plasma cells. But because these produce autoantibodies, and at least in mice, when you transfer plasma cells from a mouse with lupus into a healthy mouse, you see that this mouse also develops signs of lupus in mice. So you can show that these plasma cells are directly pathogenic in SLE. And as I said, their longevity, and also that they don't proliferate, that they don't express CD20 anymore, it makes them very hard to deplete. So when you take SLE patients and you give them rituximab, which is an antibody against CD20, and cyclophosphamide, which is a chemotherapy agent that basically kills all dividing cells, you see that even after multiple cycles, there's some rest of antibody titers that you cannot get rid of by targeting these cells. And that is the contribution of long-lived plasma cells. And we believe that this is the reason that you can treat SLE and many other autoimmune diseases, but it's not possible to actually cure them, or in most cases not possible, I should say. Because the work of at the DFZ, and this was conducted by my thesis advisor Tobias Alexander, showed that if you do a stem cell transplantation in SLE patients, you can actually, in this combination of intense chemotherapy and then replenishing the immune system, you can deplete the pathogenic memory, and by that you can, for all intents and purposes, cure SLE patients, because there are some follow-up periods of multiple years where people did not have any manifestations of the disease and did not require any more treatment. However, stem cell transplantation is, of course, a procedure that is associated with significant morbidity and mortality. So that's not something you would do to just every SLE patient. So new targets for plasma cell depletion are needed, and here are some of them that have been tried. There are, for example, from Berlin, also experiences with the proteasome inhibitors, which target plasma cells. However, these are associated also with a lot of toxicities. So what we focus on is CD38 and the anti-CD38 treatment. So before we started looking at CD38 as a treatment agent for targeting plasma cells in particular, we wanted to see where CD38 is expressed in SLE, because it's a surface enzyme that is expressed on a lot of different immune subsets and not just on plasma cells. First of all, looking at B cells, we found that CD38 is higher expressed in plasmablasts in the peripheral blood in SLE on a comparable level to patients with multiple myeloma, so patients who have a malignant disease of plasma cells. And also we found some increases in the CD38 expression in mature B cells. But as I said, all sorts of immune cells actually express CD38. And one interesting difference that we found is this difference on PDCs, on plasmacytoid dendritic cells, because plasmacytoid dendritic cells are the main producers of the type 1 interferon, the sort of cytokine I told you earlier about, which is one of the hallmark cytokines of SLE pathogenesis. And looking further, we also identified large increases in the percentage of memory T cells that were CD38 positive. And so you might say that's all very interesting, but how do we know if that's actually involved in the organ damage? And so we had the opportunity to look at the urine of patients with lupus nephritis, where also some T cells can be found. And so within the inflamed organ, we found that even more T cells were CD38 positive, which indicates that these T cells are actually involved in organ inflammation. And that's very nice, because the advantage with CD38 is that there's already a licensed drug. So if you have a new target, the very difficult part is establishing that the therapeutic agent is actually safe to use in people. And there is this antibody Daratumumab, which is targeted in SLE, which is targeting CD38. Is already licensed for use in multiple myeloma, the plasma cell malignant disease, and it can deplete CD38 high cells by all sorts of different mechanisms of antibody binding. So to summarize this first part, what we actually want to see when we employ an anti-CD38 agent is, first of all, of course, a clinical benefit. It does not matter if we deplete the plasma cells and the patient does not feel better, and if the patient feels better but we don't understand why, then that's also fine. But so that's always, to me as a physician, the most important thing. But also we expect to see depletion of these long-lived plasma cells that I told you about that produce the autoantibodies. From the data I showed you before, we would also hope that we can deplete plasmacytoid dendritic cells and thereby deplete the production of type 1 interferons. And thirdly, to deplete these memory T cells that express CD38 and that are found in the inflamed organs. And so what we did for our study is that we gave daratumumab to 2 patients with SLE. And so two patients had 4 weekly infusions of daratumumab, and then we followed them up for 1 year. And after day 120, these also received daily subcutaneous doses of belimumab, which is another antibody against the B cell activating factor BAFF. And BAFF is involved in the generation of new plasma cells and sort of lowering the threshold to generate new plasma cells. So we thought that this would be a good combination in order to first deplete plasma cells and then also inhibit the regeneration. And we had 2 patients, both of whom had 15 and more years of SLE disease and had high disease activity despite licensed and experimental treatment. And patient number 1 had especially this nephritis, the kidney inflammation that was very threatening for her, and also the inflammation of the pericardium, the lining around the heart. Patient number 2 had more of a vasculitis, which means the inflammation of the blood vessels, and also an autoimmune hemolytic anemia, where the red blood cells are targeted by the immune system. And so, as I said, the most important thing is the clinical benefit that we see. And you can see here the SLE, which is the Lupus Disease Activity Index. And you can see here after 3 weeks, so right at the day of the fourth dose of daratumumab, there was in both patients already a decrease in the disease activity, and the disease activity continued to decrease up to 2 months, and then stayed much lower than baseline in both patients up to the follow-up of 1 year. So we thought clinically this is a very, very good result. And this is again illustrated in patient 1 by the urine protein excretion. So this is a marker of the kidney damage. If your kidney is damaged, you just excrete a lot of extra protein, and you can see here all the different treatments this patient received before. And at some point, this nephritis started, and the urine excretion of protein increased and increased up to the point of daratumumab, which induced a very rapid reduction in urinary protein excretion. In this patient here, this is a more zigzaggy picture, but this is a hemoglobin, the transport protein for oxygen in red blood cells. And as I said, she has autoimmune hemolytic anemia, and you can see here she required a lot of transfusions, and despite multiple treatments, never had a negative direct antigen test. So the test that tells you there's actually autoimmune hemolysis going on. However, after daratumumab, for the first time, she had a negative direct antigen test, and also her hemoglobin levels recovered very nicely. Yes. In patient 1, we also saw an improvement of the pericarditis. Maybe you can see here that there's this pericardial effusion, this black space that is not seen at the follow-up. In patient 2, we saw here, for example, a vasculitic lesion. So this is what's happening when the small blood vessels are inflamed, and you can see here that this after half a year had healed up very nicely. But what about the immunological response? So we went out to deplete plasma cells, right? And so as you can see here, in both patients, you see approximately a 50% reduction or even more in the titers for double-stranded DNA antibodies. And this was then maintained over the whole follow-up period. However, double-stranded DNA antibodies are also produced by more short-lived plasma cells. So a good control for that is looking at vaccination titers. If you're vaccinated, for example, against tetanus, then most of the antibodies that you have in your blood are derived from long-lived plasma cells, and you can see here depletion again of at least 50% of the tetanus titers. So we would say we reached our goal of depleting approximately 50% of the plasma cells of these patients. Looking at the plasmacytoid dendritic cells and the interferon, we saw that the plasmacytoid dendritic cell counts went down early after daratumumab. However, they zigzagged up and down again, and the peripheral blood PDC count is not really a good marker for PDCs, because in active SLE, PDCs actually invade the inflamed organs and their levels are reduced. So we looked at Cyclate-1, which is a marker for interferon activity in SLE patients, and you can see that this also decreased very nicely and sustainably in both patients. So what about the T cells? We see that the whole T cell counts on the left were pretty stable in patient 1 and patient 2. However, the levels of CD38 positive T cells were reduced dramatically directly after daratumumab and recovered then over time. However, that does not necessarily mean that we did deplete the cells, because they could also just lose the expression of CD38 and still live on. And so what we did to find that out is to do single cell sequencing of the memory T cells and T cell receptors from one of these patients at 3 time points, where we can use the T cell receptor as a sort of barcode to trace these cells and then find them again. And what we found is that in the CD38 positive cells at baseline, approximately 50% of these cells have one of just 10 T cell receptor clones on CD8 T cells. And so this is very nice, because then we can see if we, after the treatment, find the same T cell receptor clones again. And in the CD38 negative cells, they were much rarer. So looking at the follow-up time points, we found that the fraction of the CD8 memory T cells that had one of these 10 TCR clones is actually increased. And so it seems that we did not actually deplete these T cells, because they were there at the same frequencies as before. However, that does not mean that daratumumab did not have any effect on T cells, because we can look at the expression of different cells, of different genes in these cells. We can see that there was a reduction in the expression of genes that are associated with T cell activation and also with type 1 interferon activity. At the same time, we saw a bit of a recovery of genes that are associated with cytotoxicity, because a recent report said that CD38 positive T cells in SLE are actually associated with increased risk for infection, because these have lost their cytotoxic ability. And so we think that's also a positive effect of the treatment. So to sum things up, we saw the treatment of 2 SLE patients with daratumumab, and I want to be very clear that this is very nice anecdotal data, but this does not constitute a clinical trial. But in these 2 patients, we saw a large clinical benefit that was sustained over a whole year and only very limited side effects. We saw a depletion of the long-lived plasma cells, or at least we saw indirect proof of that. We saw we definitely saw a reduction in type 1 interferon activity, maybe induced by the depletion of plasmacytoid dendritic cells. However, we did not deplete these memory CD38 positive T cells, but we did induce some modulation in them that we think is beneficial. So in summary, that went pretty well, and we hope to follow this up with more patients in the future. One last point is that the targeting of CD38 is not a concept that is only valid for lupus or for rheumatic diseases, because there are a lot of autoantibody driven diseases. And so here in Berlin, we together with the neurologist, we treated one patient with an autoimmune encephalitis, so an inflammation of the brain, who had this anti-Casper2 antibody, and this patient received 12 cycles of daratumumab, and also in this patient, daratumumab was associated with both a reduction in autoantibodies and an improved functional status. Cool. And with that, I have to thank a lot of people, because as you all know, science is a team sport. And first of all, I want to say a huge thank you to Tobias Alexander and Falkipe, who supervised me at this work. I want to thank people like Marie and Henrik and the team of Pasi Mashregi, and also the medical doctors who took clinically care of these patients for the great collaboration, the whole team of, or the whole working group of AGHIPE. And I also, as Andreas said, also on the side doing a few different things. And there I want to say thank you to Anja and Raluca and Helena for sort of adopting me when I thought I don't have space or anything. And also want to say thank you to Dominic and Dimi and Ronja for being great support and great friends. Thank you very much.